the University of Arizona Bio5 Institute, we bring together hundreds of multifaceted experts that include world-class bioscientists, engineers, physicians, and computational researchers. This team science approach is designed to ignite creative solutions to the many complex biological challenges facing our families and communities, and has resulted in disease prevention strategies, promising new therapies, innovative diagnostics and devices, and improved food sustainability.
Hello, welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's Bio5 Institute. My name is Brittany Ulorn.
And I'm Amy Randall Barber.
Certain diseases like Alzheimer's and high blood pressure are known to alter the link between brain activity and blood flow changes into the brain, leading to improper blood flow delivery to our brain cells. So this eventually leads to the death of brain cells and cognitive decline. Today, we're joined by Dr. Paulo Pires, Assistant Professor of Physiology and Surgery and Bio5 member. Dr. Pires studies how blood flow to the brain changes between healthy and diseased states, with an eye on possible therapies that can improve blood flow to mitigate disease. So thank you so much for joining us today.
Oh, thank you. I appreciate your opportunity.
Of course, you're welcome. So I was wondering if you could tell us a bit about blood and the brain. So why is getting, an adequate amount of blood to the brain so important? And then how do different diseases like Alzheimer's affect it?
So the brain is very interesting in that aspect because it's a very high active organ. It requires a lot of energy in order to operate. Like we all know, we're all thinking all the time, we're listening. I mean, everything that we do is somewhat controlled by our brain. And one of the paradoxes of the brain is that even though it requires so much energy, it doesn't store anything. Like there is very minimal storage in the brain. So for example, if you think about when you go exercise, for example, even like most people like me, we usually wake up, we drink coffee and we go exercise, and our body can run on storage, and our muscles, they have systems inside the muscle cells that they can access glucose quickly if needed. The brain doesn't. So everything that the brain needs, the brain cells need, or most, needs to be delivered in real time. So when you have an increase in activity, for example, right now, if you're listening to me, paying attention, certain areas of your brain are processing all of this information. Those cells, if they were quiet before, now they're active. They need more energy in order to operate. And that energy needs to be delivered in real time by the blood. So whenever we see that increase in activity, there is a matching blood flow response. This is known for a long, long time. The first studies came out in the late 1800s in England. But really, it's only in the last maybe 30, 40 years that with advances in imaging, they were actually able to understand how this process is controlled. And now that we have a better understanding of how it is controlled, we're also trying to understand why it's not working properly in some diseases. One of them are the diseases that are linked to dementia, for example, Alzheimer's. One of the things that we know in Alzheimer's disease is that there is this impaired blood flow response. Now, who comes first, chicken and the egg? That's the question, right? From my laboratory, we approach this from a very vascular centric way. So we think that really what's happening in those diseases that lead to dementia is that you have an impairment in the delivery first. So your delivery doesn't work as properly. So because it doesn't work as properly, your neurons are now, or your brain cells are now active. They are not receiving as much nutrients as they need, and over time, they start to die. So you start to have neurodegeneration. So there are other sides that will come into play into that as well, but that's really where we approach from our laboratory. We think that we are going to start seeing those vascular changes first that will then lead to neurodegeneration. It's not only, right? I'm not here claiming that it's all the blood vessels, because, I know many colleagues that would be mad at me with that. But all I'm saying is that the vasculature is still central. Because if there is no delivery, then the neurons are going to get hungry and they're not going to have the food that they need. Make sense?
A lot of sense. Yes, I'm curious. You mentioned that you think something that contributes to these neurodegenerative diseases is that, when the blood delivery is impaired. So I'm just wondering, do you all know, or are you working towards figuring out what causes that blood delivery to decrease?
So in a nutshell, do we know? No. We know some, right? The whole picture we still don't understand. So one of the things that we know, so the blood vessels in our body, they have many different layers of different types of cells. One of those types of cells are called the endothelial cells. So those are the cells that are on the inside of the blood vessels. So they're lining the inside of the blood vessels, and they are in direct contact with the blood. What we know is that these cells are extremely important for understanding the environment and mounting a response that will lead to an increase in blood flow to certain areas that are active. One of the things that we know in Alzheimer's disease, for example, but that's not only for Alzheimer's, others, hypertension will cause that. Obesity, diabetes, all of these metabolic diseases, to some extent, they change the function of these endothelial cells. Now, these endothelial cells, they are no longer able to mount a very robust response to cause an increase in the diameter or the radius of those vessels to allow more blood to flush in on those areas that are needed. So that is one of the things that happen. Another thing that we know happens, and this is very particular for Alzheimer's disease, is that one of the misfolded proteins, or one of the culprits of Alzheimer's disease, amyloid beta, and it's amyloid beta is being talked a lot about in the media now with the new antibody therapies that are coming out that are targeting this particular protein. So there is evidence that amyloid beta actually causes death of other cell types in the vasculature, the ones that are responsible for changing the diameter, that are in contact or communicating with the endothelial cells. And when they die, then you lose that ability to increase your diameter. And then the amyloid forms what is called a scar, an amyloid scar in the vessel wall. But it's something, if you think like this, is like you're replacing a rubber band with a boulder, with a ring of steel. So now that ability that your vessels have to increase the diameter or decrease the diameter to adjust the needs of that particular area of the brain is lost, because now you pretty much have a rigid pipe that cannot move. So it's really, and there are other things. We know that it alters communications of brain cells with the vasculature. We know it changes some other cells that are near capillaries. It changes a lot. But most of the studies really are focused on the endothelial cells, because those are the ones, at least in our laboratory, that's our main focus, is looking at those endothelial cells. Let's see what's wrong, and can we use that as a target and maybe improve and maybe reduce or at least slow down the progression of neurodegeneration.
Yeah, that makes a lot of sense. I like that analogy you used about, how before the blood vessel was a rubber band with, all that elasticity, and then it becomes this steel pipe. And I can see how that would really, affect the blood flow to the brain.
Yeah, and there are other, yeah, go ahead.
No, go ahead.
No, I was going to say there are other implications on that as well. I mean, I will try to not get too technical about it, but one of the things that we know as well is that this elasticity, if you want to call, like in the term that is used in the literature, is pulsatility, because these vessels, they really have a rhythm where they go up and down in diameter. And that pulsatility is also being shown to be important not only for the delivery, but also for removing a lot of these bad proteins that are in the brain parenchyma. So now when you have that pulsatility, normally kind of generates a flow of fluid that kind of washes, like a washing machine, just washing bad things out of your clothes or your dishes. Now you lose that. So not only you're preventing proper delivery, but you're also losing that effect that will wash away all of those bad proteins. So you create an environment where not only your brain cells are not receiving the nutrients that they need, but what needs to go out, what needs to be cleared out, is now staying. So you're generating a toxic environment for those brain cells.
Really quite an environment to start disease or to further disease that's already, underlying, that's already there. So yeah, big role that blood flow has to the brain.
Yes.
Yeah, definitely. And so you mentioned Alzheimer's and dementia. Are there other neurodegenerative diseases that you're looking at are included in your work?
So yes, we have some that we start working now, and some that we have plans. So one of them and this comes from my PhD training at Michigan State University. I worked a lot with ischemic strokes. So now, and ischemic stroke is a vascular event. Right? There is no more vascular disease in the brain than an ischemic stroke. So we're also, some of the studies that we're doing, we're inducing this focal ischemia in the brain, and we're looking at different targets, different proteins that we may be able to access and say, okay, if we activate this, can we make the infarct or the loss of brain tissue smaller? Can we improve recovery? Can we reduce the loss of motor coordination, for example? Those are not as much dementia as we think about dementia, because when people start thinking about dementia, it's mostly you start thinking about, oh, you're losing your memory, you don't recognize where you are anymore. But there are other parts of dementia, which is called it's a subjective cognitive decline, that also involve motor function, like fine motor skills. I think about that with my grandma. She loved to knit. And then when she started aging, she started losing that ability to actually knit. And that is one part of dementia that we don't think about. And that is one that is linked to stroke, because most strokes are going to affect motor regions rather than memory regions, at least the smaller strokes that are not massive. So we now have a new model to induce strokes in our mice, that instead of causing a damage, that if you go in the scientific literature, most of the studies use a model that was developed in the 90s, where you go and you block one of the major arteries on one side of the brain. So what happens is you end up with this massive damage of almost 50% of your brain is gone. We're now doing a much more focused stroke, where we actually activate a drug with light, and we can choose where in the brain we can actually cause that damage. And by changing the size of the light that we're adding, we can make the stroke bigger or smaller. So we can look at different aspects and how different areas of the brain may be responding differently. There are different targets that are for strokes here, or strokes there, or the vasculature itself. So that is another one. We have plans of looking at other diseases such as Parkinson, Huntington's. The evidence for a vascular impairment in those diseases is not as strong as it is for Alzheimer's, for example, but it's still there. And so, we're slowly trying to expand our portfolio and just see what is happening with the vasculature in all of these different kinds of situations.
Yeah, that's really awesome. And then you may end up finding similarities in some of the different diseases, I would imagine. Yeah.
It's very likely that there will be. I mean, there is one of the things that it's interesting about the blood vessels is that there is a lot of redundancy in the system. Mm-hmm. So you have many different pathways that will culminate in the same response. So, sometimes one is down, the other one just goes up to try to overcome the loss of one. But there are some common themes that at one point you reach the end of the funnel. And if the problem is right there at that end of the funnel, then no matter how things are working upstream, when you get there, you're just not going to go. It's like if you think about a 10-lane highway just merging into a one-way street, right? If there's an accident right there, there's nothing you can do. You're just going to be blocked. You're just going to be in traffic forever.
Yeah. I'm really curious, how did you get into this line of work? What drew you to it?
Oh, that's a long story.
Oh,
So I love to tell stories, right? Like I am even though I feel myself like a grandpa, because I just love to sit people down. Let me tell you a story. So when I came to the United States in 2007, and I had finished my master's degree back in Brazil, that's where I'm from. And I was working in cancer research at the time. And I came in with my wife. My wife got a PhD position to go into a PhD program at Michigan State University. And I was unemployed, had just finished my master's, didn't have what to do. I said, yeah, let's go, let's try something new. And when I got here, I applied to a position at Michigan State University for a lab technician. And I didn't know what the position was about, but I just applied. And as it turns out, the PI that needed that lab technician, she was working in the cerebral circulation. Completely different from what I was working before. I didn't have any idea about, I had an idea of the vasculature given my bachelor's, but I didn't really understand the brain and how the vasculature in the brain is so different. But I started, and then I was doing some things. I did in my master's, I did some techniques that she wanted in her lab. So she hired me, and we just flew. Like, we got along really well. And she still is a very passionate person about her PhD, about her research. And I just fell in love with it. And I fell in love with it and never left. And then when I went to my postdoc over at the University of Nevada, Reno, that really cemented my love for the vasculature, because I went from a lab that was stroke related, that was my PhD lab, and went to a more vascular, really hardcore vascular physiology. My mentor at the University of Nevada, Reno, he's really interested in ion channels, how they, in endothelial cells and smooth muscle cells, and how they regulate the vascular function. So, I went from, we're looking at what happens during a stroke, to the really nitty gritty of how the vasculature works. And that was also in the cerebral. And then that was it for me. I said, no, this is what I want to do. Just to differentiate myself from my previous training, I decided to go more towards, let's see what happens in neurodegeneration. But those two experiences were really what cemented my love in this area.
Wow, that's really great. Thank you for sharing that. I love stories of mentorship, and that increase your passion and kind of help you along, find your way and your path.
Yeah.
Thank you.
No, they were great. And both of them, they were very supportive mentors, and they really helped me grow as a scientist.
That's wonderful. Are you a mentor to anyone currently?
I do. I have my students in the lab right now. Right now my lab is kind of small because we're transitioning in funding, and I'm in the process of hiring more people. But right now I have one postdoc, one PhD student, another one that will come back. She's on her first year, so she's doing her rotations. So she's coming back in May. One master's student and a couple of undergrad students.
Wow.
And I mentor them as much as I can. I love them, because I had great mentors. I just want to pass that along.
Yeah. Yep. That's great. Thank you for sharing.
I think we hear that from, a number of the people we've had on our podcast is, their passion for serving as a mentor right now really stems from those experiences they had when they were younger, when they were going through undergrad or grad school and having really incredible mentors, and now they want to pass that on to the next generation. So it's wonderful that you have found that as well.
Yeah, and that was one of the driving forces for me to go down the academic scientific path. Because nowadays the private sector has so many opportunities for really good science. But to me, at least from my understanding, you don't get the same type of mentorship, such close mentorship as you get in academia, where you're training someone to be a scientist. And you watch them grow and you watch them develop. It's unique and it's beautiful.
It is. I love that.
Yeah.
I'm curious, kind of going back to something you were talking about earlier. I know you were saying how your research is really rooted in understanding the biology of these diseases and understanding how impairments in blood flow affect them. How does your work help to inform potential therapies for diseases that might be, in the brain affected by blood flow?
The way that I think about this is that it's really what we were talking about a little bit before, is finding that common problem. What is it? What is the one thing that all of these pathways are going to merge into? Maybe be 2 or 3 different things, but are they all of these 10, 20 different ways of getting there? Are they getting there? And that's where they're stopping, and that's where they are impaired. So if we find these common targets, then among different types of diseases, then we have something that we can treat that can have a broader implication, right? So The one problem with the cerebral circulation is that it's very unique from the rest of the body. Some things that you will see that are true for your gut, for example, the blood vessels in your gut are not necessarily true for the blood vessels in your brain, which is good and bad. It's good because it gives us a better opportunity for more specialized therapies. It's bad because it's kind of difficult to base yourself, formulate your hypothesis based on a rationale that you get from the literature, because it not necessarily what happens on those blood vessels that other groups are studying is true for the brain. So we always have to do a little bit more groundwork. But really, the goal is we find the funnel. We find where everything is stopping, and can we intervene? Is this something that is unique enough that we can improve its function in the organs where these diseases are happening without messing with the entire physiology of the body? Because, that is really one of the problems. For example, let's think if you want to do a therapy that is going to improve the liver, right? We're going to make your blood vessels dilate, increase in diameter, more blood going into the brain. Now, you don't want to do that in your entire body, because what that's going to do is going to drop your blood pressure. And then, you may not be able to get out of bed. Yeah, your brain is doing fine, but then what else? So we need to find what is unique about the cerebral circulation, and if we can target, because another problem is getting drugs into the areas that they need to go into the brain. So, it's a fine balance, but that's really where we're gearing towards to. And the way that my lab is trying to go about this is using gene therapy. There's this study from a group, and that's what we got the Alzheimer's Foundation grant to actually study. So a group in Germany, they did a screening of many different types of viruses, and they were able to find one particular serotype of virus that is fairly specific for endothelial cells in the blood vessels in the brain. It doesn't, from all of the other organs that they studied, they couldn't find a virus expressed in those areas. So we are making use of that particular tool to start playing around with things in the brain. Okay, so because we can use that virus to add things into the endothelial cells, we can use that virus to remove things from there, right? So if we think that this protein is being bad, let's just take it off. Let's see what happens. So that's really where we are. We're using this tool that was developed and in a way of targeting more specifically. And then if you can target more specifically, maybe that can be, we can pass that on to a therapy. I won't go that far because we all know that, I'm a basic scientist, but it's a start, right? We can start playing with this. And then once you find a target, then we can start playing with drugs. Maybe we can look at pharmacology. Let's add a compound here. Let's see what happens. How does the whole animal take this? So that's our approach.
Yeah, I think that's a really great explanation. And I like what you said at the end about, you're a basic scientist, so you don't want to take it too far. But I think that's, what's important is we don't just get excited about the people doing the directly, translational projects. Like those are obviously very important, but it's good to remember that even the people understanding the basic biology and the mechanisms, like doing this basic science, it still has these large impacts that will one day, help patients. So I think it's great that you're bringing light to that and telling us about the big impacts of your work.
That's what we hope, right? Since I'm studying those neurodegenerative diseases, the goal and the hope is that we'll find something that will help these patients. At least if we are not the ones to develop, we found something that can be taken by a pharmaceutical company or by other more translational researchers, and they can make use of that in a therapeutic setting.
Well, and I think that's the beauty of the Bio5 Institute as well, is having others around you that you could, potentially collaborate with or go to for, working on a project together and creating something or getting information from someone that helps you to kind of move forward with something that you've gone and found. So we see examples of that all the time here at Bio5, and I just love hearing about them and making stories about them. So thank you for sharing that information. And
yeah, of course. Yeah, I'm new to the Bio5. I think I just recently became a member, but I do know quite a few faculty that are members, and I'm trying slowly to get to know everyone and all of the possible opportunities for collaborations and drug development and tether ligands and all those great things that you guys do at the Institute.
Yeah.
I'm curious, I know you mentioned earlier on, you have a wife who also has her doctorate. What are the types of things that you and her enjoy doing outside of work? So I think, we talk a lot on the podcast about work-life balance and how being a PI like yourself is a very difficult, demanding job. So what are the kinds of things you do to take a step back and recharge?
Okay, so I'll tell you the things we used to love to do before, the newborn came up. Because now really it's just sleeping whenever we can. But really, and that's another thing why we love Tucson. We like to go on hikes. Like we love to go here in Tucson, Andy, you just go 10, 20 minutes each way. You're in the middle of the wilderness, just doing hikes in the desert. We live nearby the Sabino Canyon Park. So that was our Saturday, Sunday morning kind of just little thing. We would go and just do some of those hikes over at the park. And that's really what we hope to get back to doing soon once the kid is a little bigger, and hopefully the pandemic goes down as well, because even though we're outside, we have a little kid that's not vaccinated, but hopefully we'll get those back again. But really it's going on hikes and eating. Like we're foodies. We like to go to restaurants and just
Tucson
eat.
is a great place for that.
It really is. Like the food here is outstanding.
Well, wonderful to hear. I think that resonated with me a lot. I have a 6-month-old right now, and I know what you mean about, used to loving to hike and get out every opportunity that my husband and I could, but now with the baby, it's just not as practical. But as he gets older, I'm sure we'll get back into, being more outdoorsy. So looking forward to it.
It's just those first few months are and you're tired, especially for you. You just went through the pregnancy and everything. You're probably way back on your sleep schedule. So
yeah, it's tiring, that's for sure. I don't know if I'll ever catch up on sleep, but I could use a few solid nights.
Probably in 18 years, I would say.
Yeah, when they go to college, when I ship them off.
Exactly.
Aww. Well, we like to end these podcasts on a fun note, even though we did just end on a lighter note, I suppose, with that question. But we do like to think of our researchers as superheroes. So if you were a superhero, what do you think your superpower would be?
So I thought hard about that question because I really don't know. Is there could be like super nagging or something like that? Because if you ask my students, they're gonna say that I nag them all the time. Come on, we need this, finish, we need this data for the grant, we need this to finish this paper. So probably super nagging would be my superpower. And theirs would be the power to block me when I'm telling them, when I'm annoying them.
Oh, they just have these helmets, I can imagine, that just kind of block out the nagging from you. That's funny.
Oh, the noise canceling headphones. I open the door to my office, they all go.
Oh, that's hilarious.
No, but yeah, that would be my superpower.
I like that. Well, thank you again, Dr. Pires, for joining us today. I know Amy and I both really enjoyed our conversation with you, and I know our listeners will get a lot out of it. So thank you again for joining us on this podcast.
Again, thank you very much for the opportunity. This has been a lot of fun, and I'm always glad to talk a little bit about our work. So thank you. I appreciate it. And I appreciate all the hard work you guys do at the Bio5 Institute.
Oh, thank you. We appreciate that. Well, thank you also to our listeners for joining us today on another episode of Science Talks. For more information about the Bio5 Institute, please visit our website at bio5.org. And from all of us at Bio5, we will see you in the next episode.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's Bio5 Institute.